on the current COVID trajectory, what do you think that would be?
You know, Mark, it would be everybody doing things consistently the same. You know, as I've mentioned so many times, one of the beauties of our country is that it's the United States of America, and the federalist approach that go back to our founding fathers give each individual state so much leeway to do things the way they want to do it, which fits well for a large country that has demographic, geographic, cultural differences, which are the beauty of the country. That works well. When you have an epidemic, a respiratory pandemic that's as efficient in spreading as this is, and has such an impact on the health of the people, you can't have one thing, one state, one region doing things one way, another one the other way, because what you do impacts the entire country. So you want to be able to be part of the process that diminishes and ultimately crushes this outbreak. And what we've had historically is that incredible disparities in how we do things. I mean, it's become masks are good or masks are no good. You know, closing the bars are good, closing the bars are not good. That doesn't work, Mark. We've got to be able to get control of this outbreak in a way that's consistent and uniform. So as a nation, that would be my recommendation. And of course, let's get it out of the political arena. Let's forget this divisiveness. Divisiveness in an approach to a public health problem is just a recipe for not getting it right.
Yeah, that makes sense. So I'm curious what you expect to see over the next few months, especially with the incoming Biden administration. And I'm curious if you have any insight into their plans, because, you know, I have to say, I'm personally encouraged about the selection of Ron Klain as chief of staff. You know, people watching this may not know he was in charge of the government's response to the Ebola outbreak, and I was quite focused at that at the time, and you know, I thought it was handled quite well. So I take it as a very good sign that someone with that experience is being elevated in this administration. But I'm curious if you have any additional insight into their plans or what this is going to mean for your work at the NIH or the work that has to get done at the CDC or the other agencies.
Well, your instincts are correct, Mark, because, you know, I had extensive experience with Ron Klain when he was the coordinator of the Ebola response during the Obama administration. And he is the chief of staff to President-elect Biden. And I know him well and his style. And he did an absolutely terrific job with Ebola. I haven't discussed in any substantial detail about what he is going to be recommending for the approach of the new administration, but I know Ron and I know his style, and he very likely will do the same sort of thing about having a coordinator and facilitating to bring out the best in each agency, but in a coordinated way. That's what he did with Ebola. I would be very surprised if he didn't do it with COVID-19, but he certainly knows what he's doing and knows the ins and outs of how to respond.
Well, you and the rest of the government have an important job ahead. I know our team at Facebook has already reached out to the incoming administration to help in the COVID response in any way that we can. I think that there will be a few important things that we can do together. We're already planning a push around authoritative information on vaccines, which is probably a good transition to the main scientific topic that I want to talk about today. Because there have recently been some very positive news on the vaccine front with, you know, multiple vaccine candidates, including the ones from some results back that show that these potential vaccines are more effective, I think, than most experts had even expected, and without significant known safety issues so far. But given that this is such an unprecedented situation, right, I know that lots of people have questions about these vaccines, and I think we should spend the majority of our time today answering a lot of those questions. So a lot of the questions that I've seen about the vaccine center on the fact that some of these vaccine candidates are fundamentally new types of vaccines and they were developed very quickly. So I'm curious what your view on that is, and maybe you could just walk us through the process of how we determine scientifically that a new type of vaccine is safe. My sense is most people may not be aware of just how rigorous this testing process is. So maybe you can take a few minutes just to walk us through that.
Yeah. Well, Mark, we should probably start off by getting people to appreciate that this has been a process that has been done extraordinarily quickly, but not at the expense of safety or of scientific integrity. So what we've seen with these new platforms, and for those not familiar with this, a vaccine type is referred to as a platform. So a live attenuated vaccine is a platform. A whole killed one is a platform. A soluble protein is a platform. The platform that you're talking about is a messenger RNA platform, which is a highly adaptable, extraordinarily promising approach to vaccines where you don't ever even need to get the virus in your hand. You can just get a gene sequence of what it is and plug it into this messenger RNA and inject it into a person, and you get the protein expressed and you have a vaccine. We adapted, we being the field, not necessarily me, but myself and my colleagues. This is the fruit of years of fundamental basic science in improving platform technology. So when people hear you're dealing with something new and you did it very quickly, were you cutting corners? Is this scientifically adequate and appropriate? Where are we going with it? Well, the first thing to ensure people that it is the fruit of years and years of incremental research that has come up with this new platform that has allowed us to do things in a matter of weeks to months that a decade ago, Mark, would have taken years to do with no sacrifice of scientific integrity or safety. So that explains the speed. What about the efficacy? The efficacy of this vaccine was proven in a phase 3 trials. And there were 2 vaccines that we're talking about, both with the same platform. The efficacy has been stunningly good. And when I say stunningly good, I mean none of us would have expected to have seen a 94, 95% efficacious vaccine with no significant severe adverse events. Not only was it efficacious in preventing clinical disease, when you look at severity of disease, and you compare the placebo with the vaccine, it was overwhelmingly favorable in preventing severe disease. So now we have a product that would have taken multiple years to develop that now show efficacy and safety to a very high degree with the potential of completely transforming this outbreak. So the natural question is, well, what do you mean by safety and efficacy? How did you prove that? Did you have pressure from the government? Did you have pressure from the company? Well, what people need to appreciate that the clinical trial process that determined this high degree of efficacy and safety was done in tens of thousands of volunteers. The Moderna trial had 30,000 people. The Pfizer trial had 44,000 people in the trial. And what was shown is that what people don't understand is that the data, when it's accumulated to determine if it works, the company doesn't get the data, the government doesn't get the data, an independent data and safety monitoring board made up of vaccinologists, immunologists, virologists, statisticians, ethicists, independently evaluate the data. And when they feel the data truly show safety and efficacy, then the company gets the data. They look at the data, analyze it, and then put in to the FDA for what's called an emergency use authorization, or ultimately a biological license application, namely approval. the career scientists of the FDA, no politicians, but people who do this every day, evaluate the data. They then look at the data together with an advisory committee that's established. It's the Vaccines and Related Biological Products Advisory Committee, referred to as VRBPAC. Another independent committee works with the FDA to determine, is it ready to do what we're going to be doing this coming month in December, starting to distribute the doses to the high priority. That entire process, Mark, is independent and transparent, because ultimately all the data that are the foundation for those decisions are going to be made public and published in a peer-reviewed journal. So I know there are understandable questions about, is there something going on to try to slip this through, for one reason or other? You can be absolutely certain that that's not the case.
Sorry. Yeah, that's all good to hear. One new type of these vaccine platforms that you mentioned is mRNA or messenger RNA vaccine, and I personally find this advance potentially very exciting, because if this new type of vaccine works as well as it seems so far, then it could be possible to build on top of this advance to develop other kinds of vaccines for future diseases a lot more quickly and effectively. But I'm curious if you can walk us through how these types of vaccines work and why this new type of approach can be such a positive development, not just in this case, but more broadly, potentially.
Well, so the way it works is that it eliminates so much of the things that had formerly been tried and true. So the standard classic way of making a vaccine is you have to isolate the pathogen in question, whatever that might be, a new virus or what have you. Then you have to grow it up efficiently in either eggs or cells. That takes a long time. It's fraught with things going wrong. You grow it up, then you purify it, and you either attenuate it to give a live attenuated vaccine, or you inactivate it to give sort of a killed vaccine. That's the standard way of doing things. Over the years, given molecular biological approaches and the advances in technologies, we've gotten to be much more sophisticated, not requiring having to grow up the virus. With messenger RNA, RNA is the molecular means of coding for proteins. So if I want to vaccinate you, Mark, with a component of the COVID-19 virus, the SARS-CoV-2, the important component is that spike protein. Because when you look at the virus under electron microscope, you have these spikes that stick out. That's the reason why you get the word coronavirus standing for crown. It looks like the spikes on a crown. Well, that particular spike protein is the thing you want the body to make an immune response against. So rather than getting involved in handling the virus, what happens as soon as the virus is discovered, the technology today allows us to get the full sequence of the virus in days as opposed to years. So then you publish the sequence, and anybody in any lab can pull the sequence out of the gene that specifically codes for the spike protein. So you don't have to touch the virus. You go in silico, in a computer, you pull out the gene and you stick it into this messenger RNA. Now, the purpose of messenger RNA in your body is to code for all the different proteins that your body makes. So now you're going to take an artificially made RNA, you're going to stick this particular sequence in, and then you inject it into the body. It goes into a cell, and it starts pumping out protein. So the body sees the protein and says, I think that's the virus because it's a spike protein. But it isn't the virus. It's a harmless protein that you made very quickly. So to give the viewers and the listeners an example On January the 9th, the Chinese put on a public database the sequence. On the 10th, we had a meeting with my team that said, we're going to take that sequence, we're going to partner with a company, Moderna, and we're going to maybe 3 or 4 days later meet and get the vaccine start. About 63 days later, 62 days later, we had enough of it to do a phase 1 trial. An absolutely world indoor record to go from the time you get a sequence to the time you have enough vaccine to do a phase 1 trial. A few months later, we were in a phase 3 trial. And then because there was so much infection in the community, a couple of months later, we started the phase 3 trial on July 27th. By the time you got to August, September, October, we had enough data so that by the time we got to November, we said, oh my goodness, we have a highly efficacious, safe vaccine that we're now going to start to administer in December. The technology that allowed that, Mark, is beyond unprecedented. It's something that if you had told me that 10 years ago when we were making vaccines for different things, I would have told you, Mark, I respect you, but there's no chance in the world that we're going to be able to do that. And it was done. And your point is an excellent point. If we're successful, which we're there, we just have to go through the motions of proving it now with large numbers of people, that we are able to really crush a disease like COVID-19 the way we did with smallpox and polio and measles. With this new technology, you're absolutely correct. It's such an adaptable technology that it would likely transform the way we look at and make vaccines.
So I've heard from a lot of people who have questions about a new type of vaccine that involves RNA or DNA, and why we can expect that this can be safe without having any issues down the line. So I'm curious to hear how you'd explain that scientifically and your thoughts on that concern overall.
Yeah, because we know what happens, from a lot of work in the animal models. So that RNA that pumps it out for a very limited period of time, enough to get a good immune response. And then the RNA degrades the way RNA generally degrades under most circumstances. So what the FDA has done is that they are not going to consider an EUA until 60 days following the time that half the people in the trial have received their last dose. And the reason the 60 days is the magic number, because when you look at the experience over decades with vaccines, the intermediate and long-term adverse events almost all occur between 30 and 45 days. So if you wait 60 days, you cushion it a little and overshoot a little. And if you don't see severe events during that time, it's extremely unlikely that you're going to see severe adverse events.
And just to clear up one point, I mean, my understanding is that these vaccines do not modify your DNA or RNA. So I think that's just an important point to clarify. If I'm getting anything wrong here, of course, correct me. But
No.
but just to make that clear.
No. First of all, DNA is inherent in your own nucleus cell. Sticking in anything foreign will ultimately get cleared. Messenger RNA is the messenger. It comes from the DNA to the RNA to the protein. Sticking an mRNA into you has absolutely nothing to do with your own DNA or your own RNA. It's going to perform its function, and then it's going to be degraded, and it's gone. It doesn't have any imprint on any genetic material of your own.
Good. Well, I'm glad we can clear that up. A number of people have also written in and asked, how do we get a clear answer on how long this immunity lasts? Right? If you're talking about, the effect going in and then potentially wearing off, does it look like this is the type of thing where we're going to need to get boosters regularly after the initial 2 doses? Or do we think that this will be effective for a long time?
Well, Mark, one of the things that we have to be honest about in science is that when you don't know the answer, you say you don't know. And I don't know how long the immunity is going to last with the vaccine. When you look at immunity to COVID, to coronaviruses in the past, they're not lifelong immunity, unlike measles, when you get infected with measles or when you get vaccinated with measles. So we would hope it's at least a few years. And if it does ultimately require a booster, fine. I think the first step, given the situation that we're in right now, is to show that it actually works. So I'm hoping that the immunity will be durable enough to get us through a couple of cycles. And if it isn't, we'll cross that bridge when we come to it and boost people.
So one question that I'm seeing in the comments are, do the vaccines from Pfizer or Johnson & Johnson, for example, have any known collateral effects or symptoms, like this person mentions a fever in adults or anything else like that?
Yeah, well, it is not uncommon, Mark, when you're dealing with vaccines, that there are immediate effects, and then there are things that are more significant that might occur later. And that's the ones we spoke about that you can capture in that 60-day period. But with any vaccine, some more than others, when you get an injection of the vaccine, you induce a response. In some people, they don't feel anything. Others feel an ache in the arm. Some may feel an ache in the arm and kind of a little chilly feeling, almost like you have a flu-like syndrome. And in a minority of people, they'll get a fever. They'll actually get a fever for a day. Almost all of this goes away within 24 or at the most 48 hours. So we have to be honest when we tell people the adverse events, if you want to call them, are low grade. They're not serious. They're not prolonged. They're low grade. But to let people think that you get vaccinated and you don't feel anything, that's not the case. You do. What your body is telling you by that response is that it's responding well to the injection. It's making the kind of response that you want.
Yeah, so one other question that I've heard a number of times is, asking why is it necessary to store some of these vaccines at such cold temperatures? And in practice, doesn't the fact that some of them need to be stored at such cold temperatures make it pretty difficult to distribute the vaccine? How do you think about that in practice?
Well, there are 6 candidate vaccines that the federal government is involved in either helping develop or supporting the trial. 5 of them now are in either phase 3 or finished phase 3, or coming to the end, not totally finished, but coming to the end of phase 3. They vary in what we call their cold chain requirements. Some of them, as you mentioned correctly, require very, very low temperatures, like minus 70 Celsius and minus 90 Fahrenheit. And others require just a plain freezer, and some require just being in the refrigerator. It varies according to the formulation. I believe that the ones that really require that very stringent cold, if they study it more, it likely would not require it. But in the development of it, they did put it under those conditions. That makes it more difficult for that particular candidate to be distributed in a country or an environment that doesn't have easy access to cold chain capabilities. But in this country, there will be, according to Operation Warp Speed, the capability of doing the kind of storing that's necessary to do that. That is the Pfizer product. The Moderna product is less stringent. The J&J product is even less stringent when it comes to cold chain, and is going to be a great deal of variability. But you're absolutely correct. Sometimes a given vaccine, as good as it may be, would have to be utilized only in a circumstance where you can handle those requirements, where other vaccines may not require as stringent a cold chain capability.
Yeah. So, one thing that that I just want to say is, and I worry about here, is that no intervention is ever going to be perfect, right? But just because something isn't perfect or may have some issues doesn't mean that it's still not significantly better than not getting it at all or not trusting it. And it seems like this is a fundamental question about science these days. And I'm curious if there's any kind of wisdom that you would want to share with people about kind of the perspective on risk benefit in thinking through this.
Well, you're onto something there, Mark, and that's something we deal with in medicine time. Because when you're dealing with either the treatment or the prevention of disease, that you're going to do interventions, and there are no interventions of anything that don't have some risk. It might be a minuscule risk, but it's nothing. It's nothing that is completely free of risk. You take an aspirin, there is a risk for some people for an aspirin. So the way we deal with it, we deal with what's called relative risk and risk-benefit relationship. So if you look at the risk, the relative risk